Gosh, that was a big mistake from me. But with that, today is about introducing you to QTLs and QTL analysis. And to keep things short and snappy, I do need to mention that today's material, this is something that I haven't created myself. However, I have tried to summarize as much as I can what the general pipeline for QTL analysis is. And I'm going to talk a little bit about the entirety of today's material in just a second. But to move on rather quickly, what the outcomes for today's are to understand what QTLs are and the file structure necessary for QTL analysis, as well as understanding the general pipeline for QTL research. The thing about QTL research is that it is so vast and actually very difficult that is not really possible to summarize it within 1 hour. And therefore, what I can do is just point you towards a direction that may be able to cover this in a better way. And that direction for sure exists because materials for today's workshops are made available by the QTL mapping carpentry style tutorial created by Susan McClatchy from the Jackson Laboratory in Bar Harbor, Maine. The tutorial itself is extremely extensive and extremely valuable. If you do want to know more about it, I'm posting a link to it into the chat, but we will have time later to go through it in more details. I do need to mention that for this tutorial, you will require the R package RQTL2, which should be pretty snappy in installation if you already know how to use R. The original content is roughly 10 hours long. What I'm going to be doing today is just to give you a general overlook about what happens within the tutorial itself. So for the ones that do not know what QTLs are, QTLs stand for Quantitative Trait Loci, and these are, for the folks that do not know, these QTLs are traits that control, excuse me, I blanked out there, but these are traits that control phenotypes that are a little bit more complex. There we go. I was looking for the word complex, such as height, yield, disease resistance, amongst others. And the QTL mapping, the process of QTL mapping is to identify these genomic regions which contain these loci of interest. Now, QTLs, in order for you to carry out QTL analysis, you require something called a marker. And markers are, for example, SNPs, or single nucleotide polymorphisms, or microsatellites. If you are new to the word SNPs, it's in the sentence or in the word itself, in the acronym, what it is. It's a single nucleotide polymorphism. My colleague here, Carlos, is a human as well. We share most of the DNA. However, perhaps in a single area of the DNA, he may have a different base than me, and that is a single nucleotide polymorphism that may be used as a marker if this specific trait is connected to a phenotype such as height, yield, or disease resistance. Eye color, hair color, so many other things, yes. So yes, markers are generally linked to a phenotype, And for right now, I want to say this is what we need to know about SNPs themselves. I did underline this one. They serve as signposts indicating the neighborhood of a QTL that influences a phenotype, in case you've never understood what a SNP is or what a QTL study is. So what I want to do now is to introduce the study design and pipeline that the very long tutorial actually carries out. We are using data from a 2015 publication from Tien and al. If you do want to read the paper, I'm going to link it into the chat once again. The paper stands for Identification of a Bile Acid Transporter, SLCO1A6, as a candidate gene that broadly affects gene expression in mouse pancreatic islets. Now, without going into too much detail of what the paper actually covers, what the paper discusses is diabetes, specifically type 2 diabetes. If you do not know, there are two types of diabetes. Type 1, which is when the immune systems attack insulin-secreting cells and prevents insulin production, and type 2, which is what is studied in the paper, where the pancreas makes less insulin and the body becomes less responsive to this insulin. Here is an image that simplifies what what type 2 diabetes actually does, or what a healthy body does. There is an insulin receptor which will take the insulin from the bloodstream and will activate a glucose transporter, which does take in sugar from the bloodstream. Or in this specific case, we have type 2 diabetes, where the receptor is desynthesized to the insulin itself, and therefore there is not going to be the transporter activated to take in sugar from the bloodstream, leading to a buildup of sugar and disease. The study itself is looking at a resistant strain of mice and a susceptible strain of mice, a susceptible strain of mice to type 2 diabetes. Now, what we are seeing here, let's just call them B6 and R, or B and R. We're seeing a couple of generations, so the first cross and then the second cross, and using the markers that we have for mice, as well as the information from the genetic information from the second cross, we can go ahead and carry out quantitative trait loci analysis, or just as we know it, QTL analysis. The mutation prevents the production of leptin, a hormone that regulates hunger and satiety. I'm not sure, once again, how to say it. When leptin levels are low, the body does not receive satiety signals and continues to feel hunger. And therefore, these leads to becoming obese. And obesity is one of the risk factors for type 2 diabetes. And what this experiment wanted to do was to use the genetic variation between the B6 and the BR strains to identify genes which influence the type 2 diabetes. I think it's wrong for me to say that this is pretty straightforward. However, recapping, we have a resistant strain mixing or crossing with a susceptible strain. We have a first generation, and what we're doing is that we're using the data from the second generation in order to find out whether there are any QTLs related to type 2 diabetes, or excuse me, leptin production. this is the parameters themselves go into more detail on the topic itself. Now, I needed to switch gears a second here because one of the issues that I've always had when I was learning QTL mapping on my own, well, what are the files looking like? And I think that is an important answer or an important question that students and researchers should ask themselves. What does a file look like? And I think giving examples of what a file, in this specific case, files look like, can really help with moving forward with the research itself. So for QTL analysis, you require, I think, 4 files. And the first is the first 4 files, or the files in general, are CSV files. And these contain IDs in rows. And in this example, you can see what each alleles are. Excuse me about that. BNR representing the founding strains of the intercross, which are BP and RR. The BNR alleles themselves represent the haplotypes inherited from the parental strains. We're not going to be using too much of the long names. Let's just call them B6 or BB and BR. The lesson itself, so the longer lesson, looks only at 3 possible genotypes. And I think it is actually enough for you to learn how to carry out QTL analysis. This is not the only CSV file that we require. We do require a genetic marker map as well as a physical map when available. Now, when you look at a genetic map, what you're seeing is the marker name, where it is found, and the location in centimorgans. If you have never heard of what a centimorgan is, centimorgans essentially is a unit for measuring genetic linkage. A genetic map is actually also called a linkage map. And essentially what it does, what a centimorgan is the distance between the marker to a potential location of a gene in a gene of interest, excuse me. And it's not always easy for me, or it's not always easy for everybody to understand what a centimorgan is. But just do know that if a centimorgan is 1, that means that during recombination, it is more likely that 2 genes, or for example, 2 markers, are, I believe, carried over at the same time. Meanwhile, 2 markers that are very far from each other will have a much higher centimorgan. Let me check my notes to make sure that that is correct. Oh, ooh, what's going on? Why are you doing that? Excuse me, I'm having a bit of an issue with Zoom. Okay. Now it works. Okay, cool. I was not able to look at my notes. I was struggling a little bit, but the higher the centimorgan, the more, I believe, the more likely the 2 markers or the two genes are going to be separated during recombination, whilst the low centimorgan, meaning that the two are likely to be moved together. And so here a centimorgan is 0.18, which is very low, meaning that they're likely going to be moving together. A high centimorgan, let's say, will be higher than 1. So 1.0, it's not necessarily low. A high centimorgan is 50. That's the highest centimorgan. That is, ideally, these two things are not close to one another. What another map that we require, or not require, if available, is a physical map, and this actually gives you the position of the marker in bases, meaning that this is an actual physical map of the chromosome itself. Not every organism out there has a physical map, and that's why, if available, is given. But if you do have access to a physical map, if you are working on humans or human genome or mice or Arabidopsis thaliana, there is physical maps out there for you to use. Then you also need the numeric phenotypes that separate. You need a phenotypic or a phenotype CSV file, and this is a file that will give you knowledge on whether a phenotype is present or not. In this specific case, agouti 10. Agouti is a type of 10 in mice, and essentially is when mice are brown. That's the easiest way for me to actually say it, and whether it is tufted. So let's look at mouse 3051. It has a 10, but it is not tufted. The next one, 3551, it has a 10 and it is tufted. So more importantly, what we're looking here is the insulin levels. So it is important to also have a phenotypic CSV file. And once again, for gene expression data, we have columns representing chromosome and physical position of genes, as well as gene ID. The covariates shown below include sex and parental grandmother. So we are actually also able to follow through the lineage as well. Lastly, you do require a YAML or a JSON. Here we have a JSON file that actually tells you what all of the files are and where to find them. And I think it's kind of wrong for me to say, and this is it. You can start your QTL analysis. I mean, looking at a laboratory, this is probably months and months of work before you get to this point. However, once you have done your work, you can go ahead and start looking at QTLs. Let's look at the pipeline just a second, and I hope that I can put this all on the screen, because this is what the pipeline for general pipeline for QTL analysis looks like. It is not straightforward, or perhaps it is not super easy to understand, but I'm gonna do my best to explain what the pipeline does. I do have notes. I think I can actually put them side by side. Let me do that. These are different notes than the ones I couldn't figure out earlier. There we go. And I can share again. I'm gonna share my entire screen this time because you can probably see you can probably see both of the notes as well as the pipeline itself. So the first thing you do is that you take the genotypes and genetic maps and calculate the genotype probabilities. This uses marker data and recombination frequencies to estimate the probability of each individual having a specific genotype at an observed location in the genomes. I tried my best. Actually, the notes are just down here, so if you get lost, you can always go down and read. I tried my best to identify what it does and why they matter. So why calculating genotype probability is important. It's important because genetic markers are not placed at every single base pair, and this step fills the gap to improve the QTL detection accuracy. Genetic maps are extremely important in that specific case, and with the genotypes, you can get the genotype probabilities. You could go ahead and perform a genome scan from here, but this is not suggested. There is something called a kinship matrix that you should also follow through with. A kinship matrix estimates the genetic relatedness between individuals based on shared alleles. Why it matters is that it corrects for kinship, helping to control the population structure, preventing false associations between markers and traits. So a genome scan can be done directly with the genotype probabilities, just that if you want to correct for kinship and make sure that you do not have any false positive, you do require a kinship matrix. Once you have this side of the tree figured out, you can continue on here on the right side, and this is where a lot of the heavy lifting is done. Taking into the genotype probabilities and the kinship matrix, you take in also the phenotypes, the covariates, and the physical map, if there is one, and perform a genome scan. What this does is that it tests for statistical associations between genetic markers and the traits of interest across the entire genome. It helps to identify potential regions linked to trait variation. The thing about it is that there can still be a lot of false positives. So there is a couple of things that you still need to do. I remember finding my first LOD peaks and being extremely happy and essentially calling it the day. LOD peaks stands for logarithm of odds, and essentially the higher the odds for a specific QTL, the more likely this specific QTL is important to this specific trait. And a high quote unquote LOD is free. So that's a good thing if you have an LOD of 3. Let's go. Why it matters, higher LOD score suggests that a QTL is likely present in the genomic region, so you want to look into the genomic region a little more. Step after that is to perform a permutation test where it randomly shuffles trait values among individuals multiple times to determine a threshold LOD score for statistical significance. It ensures that detected QTL are not just due to random chance. So essentially what we're doing here and what we've been doing so far is, hey, we looked at the genotype probabilities, and using a kinship matrix, we're trying to clean the data. We did that genome scan, we found the peaks, and once again, we're trying to clean and ensure that this QTL is quote unquote real. And once you do the permutation test, you know that you may be having some QTLs that you can work with. So what you want to do next is to estimate its effects, and you figure out whether this QTL have major or minor influence on a trait. And lastly, you focus in even more on the SNPs in the QTL to understand whether these region are what you're looking for, essentially. And obviously, this is not the end of it. You would actually have to go back to the mice and actually carry out an experiment where you do perhaps a knockout to see whether you're right or not. This is not the end of the experiment, but this is the quote unquote end to the QTL analysis. And it doesn't take little time. It's actually a really long process. What I wanted to do before I figured out that I was not recording, I had to, sorry, restart the lesson here, was to go through a couple of pages of the lecture itself, but it seems like I will probably leave that to you, or I wanted to go through 5 pages. What I'm going to do right now is that I'm just going to go full screen right here and look at the lecture itself, the Carpenter's lecture itself. It goes into a lot more details here. As I told you, I've taken this from the lecture itself, so there is no shame for me, but it does really well at pointing out what you need to do and understanding and making you understand what this specific step does. And let's look a little bit, for example, at calculating genotype probabilities. Through images, it very well establishes what you're looking for. It also explains very well what R is looking for and gives you the code that you require in order to execute QTL analysis for your own research. So not only it gives you the code, but it also gives you the files necessary in order to, you can find the files within the code itself, in order for you to carry out this lesson. So I could sit down and go through every single piece of code that they give you, but it may be a little bit redundant as they do go in and explain to you what is going on. Maybe in the future, I will make a series covering QTLs in a lot more details and actually use this code explaining what is going on. But for right now, the idea would be for you to go home and try this out yourself. What I like about this is that it doesn't only give you the code, it actually explains what is going on in each line of code, as well as the math itself. So this is something that I strongly suggest you look at in your own time, because it will be a lot better. It will do a better job than me trying to quickly run through all of this. I'm gonna go back to key points right now. And what did I want to look at? I do suggest, I'm gonna open up the linear mixed models right here, but I do suggest you refresh a little bit on your statistics, because there are a series of applications here that require understanding of, for example, linear mixed models. They do go into details explaining the linear regressions and whatnot, but I do suggest you understand a little bit of what they're trying to do here. Don't just run the code, also understand what they're trying to do. So Today was supposed to be quite short. We're actually running. I had to repeat myself a couple of times here, but I did want to stop here, and I wanted to ask whether there are questions from the audience in the room or online. And I just wanted to thank you for your time with that. But yeah, no, any questions? And if you do not have questions, once again, thank you for your time. And I do want to say this has been a learning. Oh, thank you, Erika. This has been a learning opportunity for me as well, because I do want to take the QTLs and the work that they did up in Bar Harbor and probably try to make it into a series, because there is a lot of things to learn, and I think a lot of people here at the U of A will be able to take something out of it. So with that, thank you very much. Next week, I'm going to do something similar, but with GWAS. So genome-wide association study. If you're interested in that, then I'll see you next week. Take care. Thank you, Erika.